Hi, Lex. Thanks for joining me. So we are coming to the end of the 9th year of the ADA2 Foundation, and what a year it's been. I wanted to talk with you today about the status of the potential enzyme replacement therapy called PEG ADA2, which the foundation was actually given a couple of years ago. So there was lots of work going on behind the scenes that you're involved in, and we thought that patients, families, and others who are in our network would want to know what you've been up to as the Chief Scientific Officer of the ADA2 Foundation.
Sure. First, let me give you a little bit of background because this is kind of complex. A pharmaceutical company had developed an artificial form of the enzyme ADA2 for another use. But for largely business considerations, they decided they were not going to pursue that. The company generously donated the intellectual property rights and patents to the foundation, but it's really up to us to secure the funding and partnerships necessary to actually make it a treatment that ADA2 patients can use. At this time, PEG ADA2 still needs a lot of work scientifically to prove that it is both safe and effective. We have been working on a plan to do just that.
So can you tell us what are those next steps scientifically to keep this moving forward towards an actual treatment for ADA2?
Sure. Well, we've come up with 3 buckets or areas of focused research that are needed to show that PEG ADA2 would benefit patients. The first is around the role that ADA2 plays in what a lot of patients experience, and that is inflammation. So we want to measure how well PEG ADA2 blocks the inflammatory response in ADA2 patient cells. And we can actually do that experiment in the laboratory. The second is also about inflammation, but this time specifically in the patient's blood vessels. This is what we think is causing strokes. So we hope to measure how well PEG ADA2 blocks inflammation in the cells lining the blood vessels. And again, this is another experiment that we can do in the laboratory. And finally, we hope to measure whether PEG ADA2 would stimulate patient stem cells to restore production of critical cells called neutrophils. These are cells that are missing in patients with the bone marrow failure phenotype of ADA2. We believe together, all this data will give us a better understanding of how ADA2 works in the body and ultimately provide evidence to pharma companies that PEG ADA2 is a treatment worth investing in.
So you know what you want to prove. Do you know how to do that?
Yes, we do. As you know, we have a great network of researchers from around the world, and from some of their work, we know that these experiments are not that difficult to do. We are optimistic that the funding will come soon, and we can team up with some of our researchers to conduct these experiments.
Well, that's great. So what does the timeline look like to do those preliminary studies?
Once funding is secured, we think that we can do these preliminary experiments within a year, which will really set the stage for the actual drug development work and clinical trials. This will take actually several years.
Can you tell us a little bit more about those next steps?
The next steps in the drug development process are more complex, including learning how to actually manufacture PEG ADA2 to meet rigorous FDA safety standards, running the clinical trials, and then navigating the government approval process. Fortunately, the FDA has put in place programs that can help accelerate the drug development and approval process, especially for rare conditions such as ADA2, where there is an urgent unmet medical need. But this can still take several years. However, with the right partners, we'll work to demonstrate PEG ADA2's safety and efficacy and get it into the hands of patients who urgently need it as soon as possible.
Well, I think that's a great place for us to wrap up, and we can explore those next steps maybe next year when hopefully the first round of these experiments are completed successfully. So thank you so much for taking the time to talk with us today.
You're certainly welcome.